Welcome to this introductory video by Preferred Cell Systems. Preferred Cell Systems prides itself on providing high-quality in vitro bioassays that are complete for specific applications and work right out of the box. All you have to know is what cells you want to use and what assay readout or assay output is right for you. This is usually determined by the plate reader you have available. And in most cases, if you can use a normal pipette, you can perform our assays. At Preferred Cell Systems, we have designed assays for numerous cell types. These include, but are not limited to, cells from tissues that you see here. For many of these cell types, Preferred Cell Systems offers 3 different types of instrument-based readouts. So whatever plate reader you have available, we have an assay for you. There are assays that use an absorbance or colorimetric readout based on the reduction of a tetrazolium compound to produce a soluble yellow formazan product that is measured at 490 nanometers in a plate reader. Assays that use a fluorescence output are based on the functionality of live protease in intact cells that excites at 390 to 400 nanometers and emits a fluorescence signal in a plate reader at 505 nanometers. These are particularly useful for multiplexing with flow cytometry. The majority of assays, however, use an ATP bioluminescence readout, since these demonstrate the greatest sensitivity and reliability for rare cell populations such as stem and progenitor cells. These assays require either a dedicated luminescence plate reader or a multimode plate reader. In addition to these readouts, Preferred Cell Systems also offers Methylcellulose clonal assays that use either manual microscopic enumeration of colonies or standardized clonal assays using both a microscope and a luminescence output. Let's start with detecting and measuring cells of the blood-forming or lymphohematopoietic system. The assay that first comes to mind is the Methylcellulose colony-forming cell or colony-forming unit assay. Unlike other suppliers of methylcellulose reagents, who only have the traditional 35mm Petri dish format with a very limited number of cell populations and species offerings, Preferred Cell Systems offers 3 different methylcellulose CFC or colony-forming cell formats for multiple cell populations and species. First, there's Colony Grow, which is the traditional 35mm Petri dish 1 milliliter volume format for 44 duplicate samples. If you want to save precious samples, time and money, then forget about the macro version of the CFC assay and use Cameo 4, which is a miniaturized quadruplicate 0.1 milliliter format for 50 samples. And finally, there's Cameo 96, which is the only 96-well plate fully standardized methylcellulose CFC assay format available that measures both cell proliferation and differentiation in colonies. Assays are available for up to 20 different cell populations from 8 different species. We also have another methylcellulose assay called All Colonies. This assay is available in all 3 formats, but detects very primitive lymphohematopoietic stem cells that have the ability to produce massive multilineage colonies. The colonies contain tens of thousands of cells and are so large they can be easily seen with the naked eye. In addition to the 3 different CFC assays, Preferred Cell Systems is the only company that offers non-methylcellulose, quantitative, instrument-based proliferation assay kits for lymphohematopoietic cells using either absorbance, fluorescence, or bioluminescence readouts. These are, without any doubt, the best and most advanced assays for blood-forming stem and progenitor cells you can buy. They include the hemolyte absorbance or colorimetric assays. The fluorescence assays are called HemoFluor. Multi-cell grow is similar to all colonies, but without methylcellulose, and with a fluorescence readout that can be easily combined with flow cytometry. We then have Halo Real-Time, that is used to analyze the kinetics of lymphohematopoietic growth and identify the cells in real time. And finally, the bioluminescence assays, called Halo, which are the most advanced assays for the lymphohematopoietic system ever developed. All of these assays just take a day or less to learn and are easier and faster to use than any CFC assay. In fact, all of our instrument-based assays outperform any CFC assay with respect to cell growth, functionality, and viability. This is thanks to the development of suspension expansion culture, or SEC technology, which allows normal pipettes to be used and, at the same time, increases accuracy, sensitivity, precision, reliability, and reproducibility. One other point to remember about all of our instrument-based assays is that they can be easily multiplexed or combined with other assay readouts, such as flow cytometry, to obtain the most information from a single sample. Our assays for stem cell, basic, and veterinary research consist of HemoLite, HemoFluor, multi-cell grow, Halo Real-Time, and Halo. Most of these research assays were designed to detect and quantitatively measure up to 9 different stem cell populations and 13 different progenitor cell populations from 9 different species. Preferred Cell Systems is the only life sciences company that offers assays that have been designed specifically for hematopoietic cell therapy to provide physicians and patients alike with cellular therapy product and response information that the CFC assay could never provide. These assays range from Halo PCA that can replace the traditional macro CFC assay to quality and potency assays such as Halo QC and Halo SC-IPS, and assays to assess time to engraftment and patient monitoring for reconstitution after stem cell transplantation. Finally, for bone marrow toxicity and stem cell hematotoxicity applications, a wide variety of HaloTox HT assays are available for the primary 5 species normally tested, namely human, primate, dog, rat, and mouse. HaloTox HT assays are not only available with 96 well plates, but also with 384 well plates upon request. HaloTox HT are the only bone marrow and stem cell hematotoxicity assays with high throughput toxicity screening capability. Closely associated with the assays for hematopoietic cells are those for lymphopoietic and immune cells. We have recently developed assays specifically for early T and B lymphopoietic cell populations. For mature T and B cell response studies, we've developed the immunoassays that are also available with the 3 different biochemical readouts, instead of such readouts as tritiated thymidine, radioactive chromium, BUDR, WST1, or CFSE that are often used to measure immune cell response. These new assays include the TCP or T-cell proliferation assays and the BCP or B-cell proliferation assays. These assays are available for each of the 3 different readouts and can be multiplexed or combined with other readouts such as flow cytometry to obtain the most information from a single sample. In addition to these TCP and BCP cell assays, we also offer MLC or mixed lymphocyte culture assays for 1- or 2-way mixed lymphocyte reactions. In this case, the stimulating cells are not irradiated but are treated with mitomycin C included in the kits to inhibit cell proliferation. Like the HaloTox HT for hematotoxicity assays, ImmunoGlow-Tox HT, ImmunoGlow-TCP HT, and ImmunoGlow-BCP HT are available for in vitro immunotoxicity testing in 96 or 384 well plate formats for high throughput capability. All of these assays contain and use ImmunoGrow medium developed by Preferred Cell Systems. There are many centers worldwide that are involved in cell expansion of hematopoietic and lymphopoietic or immune cells for various applications. CellExpand is a ready-to-use reagent available to expand multiple cell populations of different hematopoietic and lymphopoietic lineages. One of the components in CellExpand is either HemoGrow medium for hematopoietic cells or ImmunoGrow medium for the lymphoid cells. These media are also part of the assay kits and can be obtained separately in 100 ml or 500 ml volumes and are available in serum-free or low serum formulations. Regenerative medicine and research into the mesenchymal stem cell system is another application area developed by Preferred Cell Systems. MSCs can be used to study osteogenesis, chondrogenesis, adipogenesis, myogenesis, the skin, and stroma, without which hematopoiesis in the bone marrow would not function. The role of MSCs in the immune response is also of particular importance. To help study this system, Preferred Cell Systems has developed several research assay kits, all of which can be combined or multiplexed with other assay readouts such as flow cytometry for more information from a single sample. For regenerative medicine purposes, there's MSC Glow QC and MSC Glow SCIPS. The former is used to ensure MSC quality before and after different procedures are used, and MSC Glow SCIPS is used to measure MSC identity, purity, and strength, also called potency. Finally, for toxicity testing of the MSC system, MSC Glow-Tox HT is also available in either 96 or 384 well formats for high-throughput screening applications. And none of these assays would work without the proper growth media. All of these assay kits are available with either Crux-Rufer, human platelet lysate, or high-performance MSC Grow media with different formulations, such as low serum, serum-free, or humanized. There are also specialized differentiation media for different cell types. The most flexible assays produced by Preferred Cell Systems are the STEMAssays. These assays allow the investigator to use their own growth media and protocols and are available for either adherent or non-adherent cell types. STEMAssays can be used for a large variety of biological systems and cell types, as you see here. The STEMassay family includes STEMglow and STEMglow Real-Time, each with a bioluminescence output, STEMfluor with a fluorescence output, and STEMlight with a colorimetric readout. There is also STEMclone, which can be used as a standardized clonal proliferation and differentiation assay for primary stem cells, stem cell lines, and other cells that can be studied using colony formation. Within the STEMassay family, there is a cellular potency assay called STEMglow SCIPS that can be adapted to many different cell types used in cellular therapy, immunotherapy, or regenerative medicine. There are also specialized in vitro toxicity assay kits for high-throughput screening and testing. These include HepatoGlow-Tox HT, NeuroGlow-Tox HT, CardioGlow-Tox HT, RenalGlow-Tox HT, and SkinGlow-Tox HT. Like all of our other instrument-based assays, all the STEM assays can be multiplexed or combined with other assay readouts. For more information, please contact Preferred Cell Systems to discuss the application you want to perform. we'd be happy to help you. Thank you for taking the time to view this video. For videos on how to perform our assays, please go to the Preferred Cell Systems website and pull down the Resources tab. If you need help in finding the correct product for your application, please contact us. We hope to have provided you with information that will further your work by obtaining the highest quality results.